Kia ora, and thank you very much for giving me the opportunity to give you all an update. Ko Monica tōku ingoa, no America hau, but ko poneki tōku kainga inaiane. And what so I'm a microbiologist working on the microbe that causes Kauri dieback, and my research has been supported by the challenge. So I guess I'm going to take this opportunity to give you guys a bit of an update, and also talk a little bit about the connections that the challenge has helped me make, and how the connections, I think, can give us all a lot of hope about where we can go from where we are now. And I guess the first thing I thought I'd mention is, why is Kauri dieback disease such a difficult problem to solve? And I get this a lot, and I think there's obviously possibly some political and structural issues as to why we haven't made more progress. I'm just going to focus on the science, because I only have 5 minutes, and I feel like the politics would be all day. But this is the life cycle of the pathogen that causes Kauri dieback disease. And one of the reasons why this has been such a hard problem to solve is that it has a really complex life cycle. So at the bottom, it talks about mycelial growth, and that's basically like the mold you might find growing on your fruit bowl at home. It looks fuzzy, it helps the microbe feed. But Phytophthora, unlike a fungus or a mold, has these other stages as well. So it makes Zoospores, and these can't move around the environment on their own, but we or pigs can transport them, and these are basically bomb-proof. So they're survival spores. They can live out in the environment for years, they can survive heat, they can survive cold, and so they are really hard to get rid of. And then the other part that's a really big problem is Phytophthora can make these things called Zoospores, and these can actually move around on their own in the environment, and so these are motile spores that are specialized for dispersal. And this has been, I guess, a really big part of my research and where we started with this project. And so what I'm showing here is a microscopy image of the zoospores sort of in action. So you can see them swimming around. What you see in the background are the threads of the mycelia, and also some of the empty and full pods, sporangia, that they've popped out of. And so this is obviously zoomed in because it's a microscopy image, but this is real time. And so what you can see is that the movie has stopped. What you used to be able to see was that the zoospores swim around quite rapidly in the environment. And I guess one of the things my research looks at a lot is how microbes decide which way to go. Are they just swimming around randomly hoping they bump into a Kauri root, or is there something directed? Are they able to navigate the environment? And when I first tried to get this project funded, I think it was often referred to as a crazy idea. And so I've often been really grateful that the Science Challenge was willing to take a chance on this crazy idea. And so they funded me to look at what are the chemical signals in the environment that the zoospores are using to navigate. And one of the things I've really appreciated with this project is the connections they've helped me make with Mātauranga Māori knowledge holders, because that's been a huge part of our research. And all of the results I'm going to show you are completely dependent on their generosity with their knowledge and their time and their passion. And it's been a really fun part of the project. So I definitely want to acknowledge Matua Kevin, because he's been really great ever since the beginning, and also Ria. And so Matua Kevin and Ria have helped us select the plants that we're going to look at. And then Amanda Black comes in on the other side of the project as a soil scientist who'll help us take it from a very in vitro lab-based test to something a little bit more realistic to the field. And so I guess combining all of this together, the Mātauranga plus the microbiology approaches, what we found is that there is a really strong attraction by Kauri trees. And that might seem like a no-brainer, but it was actually not a given. And what we're doing right now is trying to find the Kauri-specific signal. There is also a generalized response that we see as well. Other plants can be attractants, others are repellents, and this raises a whole heap of research questions. But the other thing, and if you could start the video, in some of the assays we do, what you can see on the capillary on the far end are healthy zoospores that have germinated, so they stick out a germ tube. What you see in the middle is zoospores that look a little bit mottled, like they're dead, and what you can see on this movie is a zoospore has swum into the end of a capillary in one of our tests, and what you can see is it rapidly dies. So what we were originally looking for was how are these zoospores moving, and what we found out was that these native plants are sometimes actually killing them. And so this has led to aligned research, which is basically still combining Mātauranga with molecular approaches, but now broadening out our research to all of the phases of the life cycle of Phytophthora and seeing how these native plants might be used or how they might already be inhibiting or killing the spores. And I think that's a key point, is we actually need treatments that are effective against all the life cycle stages, so not just the zoospores. And so I just want to acknowledge, I can't acknowledge everybody that's helped me with this research, but these are some of the key collaborators that I work with. I just want to say our work is still in progress, but I think we have shown that native plants produce chemical signals. They definitely can attract, repel, or kill the pathogen, and that this combination of mātauranga and has been really important to our progress. And I'm running over time. I just want to throw out there that when you work across people's different, viewpoints, there's possibly a lot of debate as to what a cure should or will look like. And those are discussions that I think people need to have, and we certainly have them a lot within our team. And acknowledge all the wonderful people in my group that do all the work. And if anyone wants to talk about an outreach project that we've also started, you can ask me during the questions.
Always leave them wanting more. If you can for a second, go backwards on that Facebook page, Why Not, huh? So that people can get another look at it. And I will definitely be dropping Zoospores in conversation just to mess with people. So thank you very much, Dr. Monica. And now could I ask Professor Phil Hulme from Lincoln University and Bioprotection Research Centre to give us your 5 minutes. Kia ora.
Thank you. I guess by this time today, you're probably thinking that most of our threats to biodiversity are occurring in the terrestrial environment. But arguably, you could say that the greatest threat to New Zealand's biodiversity is occurring in the aquatic environment. And so we mustn't forget this, and the National Science Challenge has funded a number of aquatic-focused projects. The other aspect is that we shouldn't forget, while we talk about killing mammals, that we have other pests in the environment, including weeds. And actually, in the aquatic environment, weeds pose a major threat to our biodiversity. And in this project, I want to talk a bit about the work that the National Science Challenge has funded that supports understanding why some plants are becoming much more widespread in our aquatic systems than others. And this work's been undertaken not only by myself, but also with collaborators at NIWA, Paul Champion and Danielle Clements, and particularly with MPI, the Ministry of Primary Industries, through Sam Beaumont. So I'm sure that when we talk about aquatic pest problems, you're probably all very familiar with Didymo. Now, non-native weeds have been a major problem in our freshwaters, and they impact on recreation, biodiversity, and hydroelectricity. And of course, here, Didymo was the poster child of this campaign since about 2004. So hands up who knows about Didymo? That's great. That's good. That's a good start. But actually, Didymo isn't the only problem we're facing in our fresh waters. There's a number of different plant species that are causing problems, and they've been causing problems for a very long time. Okay, so hands up who knows what hornwort looks like. Okay, now hornwort potentially is a much greater problem to our biodiversity than Didymo. And here you can see on this slide that actually since the 1950s, Lagerosyphon, Egeria, ceratophyllum, which is hornwort, have been progressively spreading from lake to lake across the country. So why is this? And the reason is, it's us. It is human beings moving around these pest plants accidentally, sometimes deliberately, and causing greater problems as they spread across into new water bodies. So what could we do? Well, part of this project was to try and understand why these plants are being moved around and how we understand the movements of those plants by human beings. So through working with the regional councils, and particularly with the coordination of the Ministry of Primary Industries, and NIWA, we developed a questionnaire that was undertaken with face-to-face interviews with about 1,300 lake users across New Zealand. And we captured details of more than 1,700 lake visits. And these questionnaires quantified the movements, awareness, and compliance of people. And what we can see is that actually, over a 2-week period, we identified a 2-week period of movement as being critical for moving live material that could spread from one lake to another lake. So actually, most people, in fact, 75%, only visited one lake in that period. So actually, the biggest risk are those people who moved from one lake to another lake during that period. But that only about 25% of lake users are in that risk category. So from the next to the next slides, really focus on that 25%. Now, in this slide, you can see our beautiful country and the movements of lake users across the country. Two things to bring to your attention. The first one is that you can see there are different coloured dots. Now, this isn't just because I like colouring the South Island and the North Island different colours, but it's the software identified two very different clusters, a North Island cluster and a South Island cluster. And what it shows is actually people in a 2-week period tend not to move between our islands, not surprising. So there's very little inter-island movement, but actually most of the movement is within islands. And there are major hubs around Rotorua Lakes and also down in Otago. And the average distance that people move between lakes is about 111 kilometres. Anglers went the furthest, about 150 kilometres, and sailors, people in sailboats, about the least, about 50 kilometres. But we did have one angler who travelled, during a 2-week period, 864 kilometres as the crow flies from one lake to another lake. So users varied considerably in their extent weeds impacted them, from 10% to about 60%. But they also varied very much in their compliance with the Check, Clean, Dry campaign. That varied from 10% to 90%. And actually, if you plot those two, you can see quite a relationship, that the more a user was actually influenced by weeds, the more likely they were to be compliant. And each of the dots reflects a different type of user. And you can see that jet boaters, although often seen as a public enemy number one, actually not only are highly impacted by weeds, but also comply pretty well. So do people who fish from boats. But down at the bottom, trampers are a bad group, but they probably don't spread weeds as much. But a big group that's probably problematic are jet ski users. So what should we do? Well, what we can understand is that only about 25% of people who are using lakes are problematic or potential spreaders. And they connect lakes over a 100-kilometre distance. Compliance reflects the extent to which they're actually impacted by those weeds. And so awareness raising should actually target weed impacts, and particularly on jet ski users. So I know most of us don't particularly like jet ski users, but if that's the case, here's another reason why you should not like them, and that is because they're probably potential spreaders of pests. Thank you very much.
Kia ora. So we're all about tiaki, we're all about protection, just not of jet ski users. Nō reira, hei kōrero tanga whakamutunga, Alby Marsh from Plant and Food Research. Tēnā koe.
Rauranga tirama, I kai whakahaere, koutou mai huhui mai nei, tēnā koutou katoa. Ko Alby Marsh tōku ingoa no tauranga moana me Ngā Tihini, Ngā iwi. My talk today is going to focus mainly around myrtle rust and the kaupapa Māori research that's been undertaken in this area currently. So a lot of you will be well aware of what myrtle rust is. Arrived here a couple of years ago, but one of the keys for Māori is that it's impacting on quite a lot of taonga species listed there, Pū, Te Kawa, Mānuka, Ramarama and Rata, but it's not just specific to those, or it's not just impacting those plant species. The other concern is that it's now a pandemic, so it is spreading rather rapidly. This is the plant that is most under threat, Ramarama, Lophometus bellata, and if nothing is done pretty soon, this runs a risk of being wiped out altogether. So a lot of our program has centred around kaupapa Māori research, trying to engage Māori and bring about better participation by Māori in this research. What we also knew is that harboured amongst our kaitiaki knowledge systems that have been brought about through historical compilation, I suppose, and that they've learnt from what has impacted on them in the past. And what we were looking to do here is look at how some of those past solutions can be modified to suit today's diseases and impacts. So first of all, what we did was we engaged, we had hui with kaitiaki around the country, especially when myrtle rust first landed here. And what we found out by conducting these hui is that a lot of our people were being ignored in the response. The other thing is that people were going onto their land and collecting seed. So what we wanted to do was address that and also share the information that we had as researchers around what we've learnt from overseas about myrtle rust and how it's impacting some of the species overseas. So, as you can see there was quite a few hui being conducted, but the other concern was, we needed to provide tools for our people to be able to become their own kaitiaki, to be able to collect their own seed. So we ran workshops around the country in association with Te Tira Whakamātaki, and it's similar to the fish and net analogy. You give a person a fish and they'll be fed for a day. If you give them a net, then they'll be able to catch fish forever after. So if you give our people the tools in which they can collect and preserve their seed, then they become the real kaitiaki of their taonga. So our courses that we ran with Te Tira Whakamātaki was designed to provide those skills that will enable them to collect their own seed, dry them off, and then in those blue drums down there in that bottom picture, those are the seed banks that were kindly donated by Kew Gardens. And so they then had the tools in which they were able to become their own kaitiaki, their own seed collectors, preservers of that seed as well. The other thing we did within our project was mapped the spread, and this was done in association with B3. And as you can see there's a lot of blue spots, and those are, what do you say there? Searched for but not found, and that was prior to September 2018, last year. The orange spots are the positive finds, and the red spots down the bottom there in Te Tauihu are the more recent finds. But what we've seen in recent years, or recently, is that we've had a drying up of the data. So we don't really have much data beyond September of last year. So again, that's a big gap for our people. And one of the issues there is that we do have kaitiaki on the ground wanting to do surveillance, wanting to look after their ngāhere, but they don't have the resources in which to do so. With the maps, we can drill down a bit deeper and a bit closer. We can go into a rohe level map and identify where the hot spots are. And if we look to overlay those maps with other data like climate, wind, all of that, we can get a better idea of the movement and the hot seasons on which the spread is advanced. The other piece that we're looking to do within this project is to connect with our Pacifica cousins, learn from their experiences, but share our experiences with them as well. And the whole idea of that is a program of co-learning, but also this co-innovation. We can design and develop solutions together using the mātauranga of each. One of the travesties with a lot of research is that as soon as the funding dries up, that's the end of the program. So we're lucky that Marge and her team at Landcare Research were successful in their recent MB bid and have funding for the next 4 years now. That's 1 year into the program. And these are the 4 areas that we're looking to research. And the good part about this diagram is that a lot of the research is interconnected. And then the RA 1.4 down there is designed to keep the relationships that we've established in the program going. That's us, kia ora.
So we have a short amount of time for questions, and luckily you've got to those already. So Phil, I'll ask you to take the mic initially with this question from Bruce. Is aquatic weed invasion correlated with pest fish invasion?
To a certain extent, they are, and they're not directly. I mean, they're driven by similar factors, and that is accessibility of lakes to introduction of fish by human beings is quite closely correlated with those lakes that are accessible to fishermen and boat users who might actually introduce those weeds accidentally.
Alby, he pātai tēnei ki a koe. Alby, this is from Mara. At the Deep South NSC, it was said that only people who truly whakapapa to Aotearoa can be kaitiaki. Do you agree with that, or is there a place for keen Pākehā to connect?
We're all kaitiaki at the end of the day because we're all responsible with looking after our piece of land, the land that we own, but the land that we share. So, yeah, we're all kaitiaki, not just Māori.
Hopefully you could hear that. We're all kaitiaki. And kia ora, tēnā koe. Someone, let's do more of that. And now, Phil, I guess I'll ask you, please, which weed was mostly identified across the various lakes that you mentioned? Any findings yet for dominance in a particular weed against others, and if so, any possible reasons?
So with water weeds, there is what's called a successional process, is that some weeds will come in, and then after a period of time, the weed is replaced by other non-native plants. But probably the most widespread weed across both islands, in North Island and South Island, would be Lagarosiphon, which is a major problem. The ecological studies haven't yet depicted what it's doing, but we know that it's colonizing the shallower areas of the lakes, as well as some of the deeper areas where many of our native species are growing. The interesting thing is, while most boaties and fishermen know something about Didymo, a much smaller proportion know about Lagarosiphon. And so although you might go to these lakes and you see signs saying, watch out for these weeds, because things like Lagarosiphon, Egeria, Hydrilla all look rather similar, they seem to blur in people's mind. And so I think there's an issue there in terms of the identification and understanding, and also they have quite long names. I think that's one of the issues that we face, is actually persuading people to understand something about these weeds, particularly things like Lagarosiphon, needs more work.
And so if we could go across to Alby, are ramarama cultivators more susceptible than real species? And if so, what are the ramifications for management? Asked Bruce.
There's probably other people in the room more qualified than me to answer that, but from my understanding is that it's because it's a hybridised program that's plant that's made it more susceptible. No, I don't know. Ramarama it does tend to be a lot more susceptible to the disease. So I suppose you can either, the management of the disease is either spray the bejesus out of it, or look at conserving the seed until a more suitable solution is found.
Kia ora. Monica, so I have a couple of questions for you. First of all, from Natalie, which was the native species that killed the zoospores, and what lessons can you offer other researchers about working with knowledge holders?
All right, well, I'd say the first question I think I get every time I give a talk, and I think it relates a lot to the second question. So I'm not going to say what the plants are at the moment, and that's because, as I hope my slides indicated, it is really collaborative work, and so it's not until we're all comfortable as a team, and we're not all there yet. And so we're working on it, and once we're sort of everybody on the team is ready, but it's not just the scientific knowledge, it is the mātauranga, and we all have to be on the same page. And I guess that would be a big lesson for other researchers maybe. I think as researchers we tend to be a little bit micromanagey sometimes, and we don't like to give up control of things. And I think we have to be a little bit more flexible and a little bit more open to giving up the control of a project and saying, actually, I'm not going to do necessarily what I want to do. We're all going to do what we want to do together.
And it's really interesting. I can see why that would be challenging, because in a whakaaro Māori, in a Māori approach, you can go, well, the mauri of that tree is doing something, and you've found a scientific way of expressing that. So I see the challenge, ka mihi o ki a koe, and how you're taking on that challenge. So I'll go now to Phil. Freshwater and marine pests are the poor cousins of terrestrial pests. How do we increase the focus, awareness on these?
This is a really interesting issue in that, 1 in 4 New Zealanders supposedly has a boat. I think only about 1 in 10 actually use it. But it seems as if we do use our waterways, freshwater and marine, quite a bit, and yet we seem to have quite much greater disregard for the quality of that environment. And I think the issue is that the biodiversity of those areas, particularly the marine, and secondly to fresh waters, is much less well understood than the terrestrial. We don't have the flagship species, perhaps, that we want to raise, like tui or saddlebacks or hihi or whatever. And I think that increasing the understanding of the value of what we have in the fresh waters is important. Now, I think this is a great area where cultural convergence is important, because the mahinga kai, tuna, even things like watercress, etc., species that are of critical importance, much more to Māori communities perhaps than to Pākehā communities, are those issues that we could start working on. The impacts of these weeds and these pests, these aquatic weeds and pests on the mahinga kai might actually be an area where we can bridge new ground.
Yes, as someone who comes from Little River and Waihora, Wairewa, kei te rongo ke au, yes, it's a huge issue and really hurtful in terms of what happens, in terms of the destruction. So could I go on to Monica and Alby? One of our last questions perhaps. Oh no, there's 4 minutes, loads of time. So feel free to put some more questions up there. How do you protect mātauranga in your work?
I think this is a really sticky question, right? And I'd say, first of all, I think it's something that the challenge has been really helpful for me to help me navigate this. The other researchers, the governance group, the Kauri Māori, like getting lots of different opinions. I guess the other thing we do is I do have lots of different collaborators, and so everything is on a case-by-case basis, and what works with one person might not work for another. But it's really tricky, and if I have time for a quick anecdote, I would say I talked to an IP attorney really early on in the project just for advice to figure out what's going on. And when I was asking them about it, they instantly thought I was trying to circumvent. And they're like, oh, you could do this and get around it and put a patent on it. I was like, no, I'm asking the opposite. Can you please give me advice on how to actually protect the knowledge? And I'm open to, I don't know if you've got a better answer. It's an ongoing issue for our research and how to share it and when.
One of the misconceptions about CRIs is that they're out there just to develop IP and sell it off, and that it can't be further from the truth, because we're quite clear now. We're quite clear now about how we approach mātauranga, and it's if a body of knowledge is introduced into a program that is part of a knowledge system that is external of our organisation, then the person that was able to contribute that piece of knowledge is the holder and the owner of that knowledge. So what he contributes, he can also take out at any time. So we're really, really clear about that. The co-development part, well, that's a different conversation. And if we're modifying a piece of knowledge or contributing to its improvement, perceived improvement, then there is a different discussion had, and it comes about, it starts by where would we be without that first piece of knowledge? Where would we be? And probably we wouldn't be able to modify anything if we didn't have that initial starting point.
Kia ora. So as a final question, I was quite interested, yes, if you don't mind, I'll get to Marcus's question to you, Phil. What is your view on boat registrations to fund aquatic biosecurity?
Right, how many boaties are in the audience? Because I don't want to be lynched just yet. Okay, so I think the idea of boat registrations, a direct tax on boaties, will be resisted. What you can do, though, mooring fees in marinas, you can use some of that money. The other thing are fees for using boat ramps. Many boat ramps you charge $5 to use your boat ramp. Why not charge $6? And that $1 needs to go into aquatic biosecurity, not into the regional council's pocket to be spent on something else. So the issue there is then what you do with that money. Now, clearly having washdown facilities near boat ramps or in marinas would be a really good idea. And so perhaps showing that the additional charges will go back into useful activities for the boat users will be a better way than simply disappearing into the pockets of the government.
And just finally, because it seems, I'm going to read this as an offer. Monica, do you need more money? What would you do next, says Bill? Just very quickly.
Yes. I guess research-wise, I mean, there's always a lot of questions, and I do think what you mentioned about co-design is really important. I mean, I think people that know me know that I always have a million things I want to do, so I think part of it would also be talking to other people to see what the most urgent needs are. What we've identified currently is the small molecules that are causing these responses, but we're not actually sure yet whether or not they're exuded by the plants into the environment. And that really affects the downstream applications, whether or not you're talking about a treatment or something like co-planting. And so that would be something that I'm particularly interested in.
As again, I would find Bill if I was you later on. Engari, tēnā koutou katoa. Thank you so much to all of our panellists.